Welcome to Bossing Biology. My name's Lucy and today we're going to be looking at the lac operon. This is in the OCR exam board under cellular control 6.1.1. Right, come on, let's go and look at what's going on inside an E. coli and the lac operon.
The word operon is a noun, and it's simply a group of genes that's under the control of the same regulatory mechanism. It's often found in prokaryotes, but it can be found in eukaryotes as well. We're now going to look at the regulatory mechanism in more detail for the lac operon. This particular operon is found in E. coli. Before we look at the mechanism of the lac operon, we first have to understand the bigger picture. When in favourable conditions, E. coli actually prefers to use glucose as its respiratory substrate. But so it can survive when glucose levels are low, it can actually use lactose as a respiratory substrate as well. But would it be cost efficient for an E. coli to be constantly producing enzymes that allows it to use lactose as a respiratory substrate? What do you think? Before we get started, let's familiarise ourselves with the main characters of the lac operon. Firstly, we've got our regulatory gene. This is the gene that codes for the repressor protein. The repressor protein is a protein that is attached to the operator region. The operator region is a segment of DNA where the repressor protein binds. We'll come on to that later. The promoter is where the RNA polymerase will join to allow for transcription to occur. And then finally, it depends which question you're looking at, but often it's written like X, Y, and Z. We will have the structural genes. These are the genes that the bacterium, in this case E. coli, is going to transcribe if lactose is present. These proteins will then allow E. coli to use lactose as a respiratory substrate. Let's look at what happens when lactose isn't present. So all the time, the regulatory gene is producing the protein, the repressor protein, which is binding to the operator region. When lactose isn't present, the repressor protein remains bound to the operator region. This is preventing RNA polymerase from binding to the promoter region. This would then prevent the transcription of the structural genes from occurring. Let's just remind ourselves of the wider picture here. We don't need these structural genes being transcribed and then translated because there is no lactose present. Therefore, you do not need the enzymes to break down lactose to be used as a respiratory substrate. The phenomena of the repressor protein preventing transcription is known as downregulation. So now let's look at what happens when lactose is present. Firstly, it binds to the repressor protein. Now this is purely synoptic. What happens when something binds to a protein? Where have you seen this before? Think back to AS when you looked at enzymes. That's right. It's exactly like a non-competitive inhibitor we see in enzymes. When the lactose binds to the repressor protein, it will change the tertiary structure, therefore meaning it can no longer bind to the operator region, exactly like we see in enzymes. So when lactose binds to the repressor protein, it makes it change its shape and structure and therefore releases itself from the operator region. This then allows RNA polymerase to bind to the promoter region, allowing transcription of the structural genes. So when all the lactose has been metabolised, there's none left to bind to the repressor protein, changing its shape and structure, so it can now bind to the operator region, preventing transcription of the structural genes again.
That's it. You've just bossed the lac operon. Well done. So if you found this useful, please press like and subscribe to my channel below. Okay, now go and spread the word of the lac operon and get your nerd on.